sit back and we'll enjoy the talk on subcellular and nanosurgery. Dr. Mazur.
Thank you. It's really a pleasure to talk here. And since we have students here, as I heard Professor Johnson introduce me, I reflected for a moment on my own career and how long I actually came to Harvard. So I thought maybe I should tell you a little bit about myself. I always knew exactly what I wanted to do. It just never worked out that way. When I was 7, my grandfather gave me a book on astronomy, and I fell in love with astronomy. I knew I was going to become an astronomer. So when I enrolled at the University of Leiden, I actually declared my major to be astronomy. And one of the very first teachers I had was Oort, Jan Oort, who Leiden was a mecca for astronomy in those days. And another instructor I had was Van der Hulst, who was sort of the father of radio astronomy. So there was no better place to be for astronomy. However, my romance with the heavens unraveled in less than 6 weeks. All of a sudden, it all became about right ascension and declinations and calculation. It was all about equations, and I thought astronomy is not for me. And I went to physics because I knew I was good in physics in high school. It took only 6 weeks to discover that physics was not that different from astronomy. But I'd already switched once, so I thought I can't switch a second time. So I plowed through my undergraduate years, not being very happy actually about what I'd chosen. But there was one aspect of my work I liked, doing research. So that convinced me to hang in there, go to graduate school. And that's when I rediscovered the beauty of doing research and discovering new things. And as the years went by, and I got to my 3rd year, and was imminent, I thought, I got to think about the rest of my career. I knew one thing for sure. I was never going to be a professor. My father was a physicist, a theoretical physicist and a professor. My mother was teaching art history at the university. I was going to do something useful with my life. So I lined up a job in industry at Philips in Eindhoven. This is going to date me when I say this, but I was going to work on the development of the compact disc. They had the compact disc was a joint venture between Sony and Philips, and I saw this huge prototype of a compact disc, a big laser disc this size, and my team's task was to shrink it to the whatever 6-inch format or so that it eventually became. So I got an offer before I obtained my PhD, and I remember coming home and showing my father the offer letter from Philips, and he congratulated me and he said, "Why didn't you go first to take a postdoc in the US?" And I'd never been in the US, so I thought, maybe that's a good idea. I can learn more about lasers. I was going to work with lasers. And then go back to Philips and do this job. So I called up Philips and said, "It's okay if I defer my acceptance for a year and to go to a postdoc in the US and learn more about optics." And they said, "Sure, that's a great idea." So I thought I had nothing to lose. I wrote to 4 places, Harvard, Stanford, Berkeley, and MIT. I got 2 offers, and Harvard was actually my #1 choice. So I went to Harvard. What can I tell you? It's been a long year. My career took on a very different path than I would ever have imagined. So, if you're still planning your career, I think the 2 lessons that I learned is it's always good to have a goal. Because, that goal kept me going in a certain direction. But it's equally important to always be ready to deviate by 90 degrees. I've done that on a number of cases, and it's really helped me. Later on in my career, in these past 10 years, I've started a number of different companies. One that is in the semiconductor industry, and 2 that are, one is a software company and one that's in education. So I find myself with 1 foot back in the corporate world, and I discovered in some ways that somehow my career path took on the right direction, because I don't think I was meant to ever be in the corporate world. It's a very different world. I think I like to nurture the human mind rather than than to push an organization to develop a product. Although that's fun too. Anyway, that was just an aside. Your introduction made me reflect on that for a moment. So I'm going to talk, and I'm going to sit from time to time. I had a really bad accident in February. I was hiking. I do a lot of mountaineering, and I took my research group, 4 of my graduate students, a colleague of mine, visiting professor, and myself, to the White Mountains, which we do every year. And frankly, I consider the White Mountains hills. Okay, I mean, I would not give them the name White Mountains or White Hills. Well, it turns out we went on an overnight hike to a hut that's open, and the second day we were a little bit adventurous. We took a longer trail. I'd done it once before, and we got stuck at night. All 6 of us had severe hypothermia. We barely made it out. And 3 of us, including me, suffered severe, if you wonder why I'm wearing these weird shoes, it's because, here they are. I had this severe frostbite on both of my feet, which is no joke. No joke. But the good news is I still have all my toes. And the last little bit of gangrene fell off today as I arrived in Bellingham. Must be the nice weather here. Anyway, so from the miracles of the human body, because I don't know how these toes of mine survived, we're going to talk about cells today, which is one of the areas my research group has worked in. And these are some of the former people whose work I'm going to be highlighting. I'm going to give you sort of an overview of, first of all, how we got into biophotonics and why would a laser group be involved in doing work that borders on biology. Eva Maxwell is my former subcellular surgeon. She developed a technique to actually do surgery on an individual cell. She now works for a biotech startup company started by another graduate student of mine. Sam is our nanoneurosurgeon. He's a postdoc at Boston University. Valeria Nuzzo was a postdoctoral researcher from France, and Alexander Heisterkamp was a postdoc from Germany. He's currently on the faculty at Hanover University, and I still collaborate very closely with him. And there's a long list of other graduate students who are responsible for the work I'm going to present here. And in particular, I want to highlight Professor Donald Ingber, who is a colleague at Harvard Medical School, who got me into this. Not on purpose, by accident, but that's, life is serendipitous, and all the good things that happened in my life were totally serendipitous. And then Professor Arvi Samuel, who is a colleague in the physics department doing biophysics, I'll mention his work later. So if you look in a bio textbook, you see all of these beautiful images, and it becomes obvious quite quickly as a physicist that our field has had a huge impact on the advances in the study of living systems. For example, this beautiful picture of red blood cells, I mean, that's not done in an optical microscope. You could never get an image like this in an optical microscope. It's the electron microscope. So all of these tools that are required to provide the images that you now find in biology textbooks, such as, this image showing the nucleus of a cell and the mitochondria. If you actually look at how you can visualize the mitochondria, and I'll show you optical microscopy of mitochondria later on. They don't look like this at all under an optical microscope. And under an electrical microscope, you see some of this structure. But through all of these physics tools, optical microscope, electron microscope, many other tools, we have been able to reconstruct this visual image of what happens in living systems like cells. Now, why would somebody who uses femtosecond laser pulses become interested in biology? What is a femtosecond laser pulse? Well, I'm an experimental physicist, so ideally I'd like to bring you to my lab to show it. The best I can do is show you a picture. But if you come to my lab, you see tables covered with optics. And basically, a femtosecond laser pulse is a pulse that is measured in femtoseconds, 10 to the minus 15 seconds. Sort of the shortest possible pulse in the visible nowadays is on the order of about 5 femtoseconds. That's a 5 femtosecond pulse. Half widths at half max is 5 femtoseconds. We'd like to, stretch half widths. It gives you the shortest time. So it's a metric that can give you the lowest record. Notice that it's only a few oscillations of the optical or the electromagnetic wave. If you look at the vertical scale, you see a pretty big picture then, 10 to the 8 volts per meter. The electric field in one of these very short pulses is very, very high. Why? Because in a sense, you squeeze the photons together. Right? A femtosecond optical pulse is in a sense a pancake of light that is only a fraction of a micron thick that travels at the speed of light. So you squeeze the photons on top of each other in the direction of propagation of the light. And if you squeeze the photons on top of each other, you get a very high electric field. If you now also focus it with a lens, so that not only do you squeeze them in the propagation direction, in the temporal sense, you also focus them in the spatial sense, you can easily get fields of 10 to the 14 volts per meter. That's enormous. If you take the Coulomb field that binds a valence electron to an atom, it's only on the order of 10 to the 10 volts per meter. So imagine your valence electron in an atom. When one of these femtosecond laser pulses comes by, the instant electromagnetic wave has a much greater value than the electric field you feel from the nucleus. Normally, when you treat electromagnetic waves, you use perturbation theory. The light that shines in this room is a perturbation to matter. When you get to those values, it's no longer a perturbation to matter. The roles are reversed. It's matter that is a perturbation to the electromagnetic field. So you basically can rip electrons off atoms. You can break chemical bonds. You can do lots of things at relatively low energy. I'll elaborate on that in just a second. But what is happening is that the interaction between light and matter when you take these short pulses is very, very different from the interaction that we normally deal with. Normally, it's matter that determines what happens to light. You see my shirt here because of the way it interacts with the light. When you take femtosecond laser pulses, it's light that determines what happens to matter, something that we normally cannot imagine because it doesn't happen. Fortunately, it doesn't happen at the intensities of light that are around us. So let me give you an example. If you take one of these pulses and propagate it through a piece of glass, as shown there, you actually have effects that are not described by the ordinary interaction of light and matter, as it is described in most introductory electromagnetism textbooks, but you get all kinds of nonlinear phenomena. For example, we tend to think of the index of refraction as a constant. Glass, 1 point, whatever it is, 5 something. Water, this, oil, that. Maybe it has some wavelength dependence. But it turns out it also has an intensity dependence. The index has a term that depends on the intensity. Now, the term that depends on the intensity is usually so small that you don't notice it. But when you get to these high intensities, you do notice it. So in the front of the laser pulse, we have low intensity, and therefore we have the normal index of refraction. At the center of the pulse, we have a very high intensity, and therefore this nonlinear term becomes important. If it's a positive term, the index will be higher. It can also be lower, but let's assume that that term is positive. What does a high index mean? A high index means a lower propagation speed. So now we have this pulse where the front and the back goes at one speed and the center goes at a lower speed. The speed I'm talking about is the phase velocity of the pulse. So basically you slow down the center of the piece. Imagine a train that has 3 cars and the middle one goes more slowly than the front and the back. You get a problem. Things are going to stretch out at the front and compress at the back, and that's exactly what happens to the spacing of the wave fronts. So we come in with a monochromatic pulse, 1 wavelength. We come out with something that is a complete continuum that has wavelengths all across the electromagnetic spectrum because of this nonlinear interaction of the light with the matter. And this phenomenon is called continuum generation or also self-phase modulation. It's very easy to show. You take one of these laser beams, you focus it into some water and outcome white light. We love to show that to visitors because it's very easy to demonstrate, and it's so unusual. I know you take white light and you break it up into spectral components using a prism, and then if you take one of these components, it stays that component no matter what you do. When you get to these high intensities, you have all of these interesting phenomena that happen. Now, the workhorse for femtosecond work with femtosecond lasers is the titanium sapphire laser, which emits pulses at 800 nanometer. And if you look at this absorption of several components that are present in tissue, like melanin and hemoglobin and water, you see that at 800 is pretty well positioned to be near a minimum in the coincides the minima of these three components. So even though you don't tend to think of skin as being transparent, it is actually fairly transparent at 800 nanometer. It's turbid in the sense that it scatters light, but the light goes through. And that's actually important because as I got engaged into biophotonics, we were actually studying the interaction of femtosecond laser pulses with transparent media. And then when I learned about the bio work, I thought, oh, I already have a lot of expertise in the interaction of short laser pulses with transparent media. We can treat most tissue as a transparent medium, so we can extend our knowledge into these devices. Now, biology has made huge advances over the past 30 years. Many techniques from physics, but also many techniques from chemistry, such as this fluorescent labeling, which a few years ago was the Nobel Prize in chemistry. You see cells here, endothelial cells, and you can see the mitochondria. Those are the little spaghetti here. You can see the nucleus in red, and you can see the actin fibers stained into a different color. This is done by inserting the gene for these fluorescent proteins in the gene sequence for that particular part of the cell, so that that particular cell makes that protein that fluoresces. Here's an in vitro fertilization, the insertion of DNA material into the nucleus of a cell. That's done mechanically by making a nanoscale or microscale, or well, between nano and microscale needle to inject the material. So one of the things I'd like to point out is that the standard biochemical tools are species selective, right? That first picture I showed, the nucleus was red, the actin fibers were another color, and so forth. I'm going to show you another technique today which deals with femtosecond surgery, which is site-specific, right? If you program cells to express this red fluorescent protein in the nucleus, then all of the nuclei will be fluorescent red. You can't target a specific cell. They'll all be red. With femtosecond laser pulses, we have that specificity to a particular spot in space. That's what we're going to exploit in this talk. So I'd like to talk a little bit about femtosecond laser directions, which is what got us into this whole area. Then I'd like to talk about subcellular surgery, and finally about neurosurgery. So let's start with femtosecond material science, which is what I was involved in before I got into biophysics. The workhorse is this titanium sapphire laser. We used to build our own lasers. I was probably the first academic lab that had a femtosecond laser. Now, you just buy them, including me. It's not worth building them anymore. But at the center is a tiny little titanium sapphire crystal that generates that radiation. The pulse, if you take a commercial laser, typically is around 50 femtosecond. And the repetition rate out of the oscillator is about 80 megahertz, which means that about every 12 nanoseconds you have one of these femtosecond laser pulses that come out. The average power is about a watt. But if you take the peak power of a single pulse, it's on the order of 10 to the 10 watt. That's a whopping big number, okay? You can power probably the city of New York with that only for a few femtoseconds, but yeah. The energy per pulse after amplification is easily a millijoule, which again is not very much, right? I mean, I drop an object and you have fractions of a joule. So a millijoule is not that much, but in photons and delivered at 12 femtosecond, it's an enormous power. The wavelength is about 800 nanometers. By the way, you can't get a millijoule and 10 to the 10 watt. That would be, sorry, you can't get a millijoule per pulse at 1 watt average power. Okay, so let's get to femtosecond materials interactions first. So almost 20 years ago, we decided to study the interaction of femtosecond laser pulses with transparent material. Transparent, the definition of transparent is not interacting with light, right? Transparent is just another word of saying is not interacting with light. So why would you look at the interaction of femtosecond laser pulses with transparent material? It's because the interaction at those high powers is very different from that of ordinary light. Ordinary light would just go through. If you take a femtosecond laser pulse, then right at that focus, the intensity is so incredibly high that you rip the electrons off, the valence electrons, off the atoms, creating a very dense plasma that has quite a bit of energy over a nanoscopic or microscopic little volume, causing damage right around that place where you focused the light. And we showed that you could write gratings and all kinds of things, and that the little pits that you form, this is a dark field microscopy image of a piece of glass where we've written basically bits in one plane. But the good thing is that with this technique, you can actually write many different planes, right? One above the other by changing the Z position of your microscope. So we showed very high data storage densities and a number of other applications in microfluidics. I'm not going to get into that because I want to get to the bio work. But the interesting thing is it's a transparent material, so the energy of the photon is not enough to promote an electron from the valence band to the conduction band. You need actually quite a few photons, so the interaction has to be non-linear. And that non-linear part is very important because if we compare linear and non-linear interaction, in linear absorption, if you focus light into a material, the absorption would start at the surface and then diminish as you go into the material. If it's non-linear, you can basically confine the absorption to the part where the intensity is high enough to be absorbed. So we get complete confinement in X, Y, and Z. So we can actually deposit energy inside a transparent material. Here are some SEM, scanning electron microscopy, and atomic force microscopy images. No, actually this is only an SEM, but the combination of the two reveals that we have tiny little cavities inside the material on the order of about 100 nanometers. That's the accuracy you get. And notice also, you can sort of see that from here, but you can see it better from other images that we took. There's no damage to the material around it. Why? Because we don't deposit much energy in it. Remember, it's the intensity that is high. If you take a tiny little bit of energy, you compress it in time, the intensity gets very large. So you rip the atoms apart at the focus, but as the energy dissipates out of that volume, it basically loses the ability to do anything. So you get exquisite control in space without damaging anything around it. Okay, so how much energy do you really need to damage a piece of glass inside? How do you determine that? One would be to basically take your pulse, put a detector, and see how much light gets through. But then you're looking for a tiny change on a very large signal, which is not very sensitive. So instead, we developed this dark field scattering technique. We take a probe beam, which we focus into the material with a microscope objective, and then we collimate it on the other side and send it towards a detector. But in front of the detector, we put an inverse iris, an inverse aperture, a block that blocks the beam. Then we bring in our femtosecond laser beam, collinear with the pump beam. And as this beam causes some changes in the sample, some of the probe light will scatter around this beam block and reach the detector. So no change, no light. Change, light. So it becomes a very sensitive way of measuring any changes in the material. Now you put the detector, you connect the detector to an oscilloscope and look at the trace. So here's the signal scattered around the beam block as a function of time resolved by the oscilloscope. When the pulse hits, you get a spike, and then the scattered light diminishes and goes back to zero it goes back to 0, which means there is no permanent damage. It's just transient at this point. This is when the pump beam was about 0.1 microjoules, so 100 nanojoules in a piece of fused silica, which is ordinary glass, basically. What is this? What causes that scattering that is temporary, not permanent? Obviously, we've not made any permanent changes in the glass. We have, however, formed a plasma. We've ripped apart a lot of atoms that ultimately recombine. So this is basically the effect of a plasma. When we turn up the energy, and therefore the power, we see a very different behavior. We still see this initial spike due to the plasma, but we see a big bump, and more importantly, we see a tail here which indicates there's permanent change. It does not go back to 0. That big transient is basically the thermal energy diffusing out, changing the index of refraction as it diffuses out. So now we have a very sensitive probe for permanent damage. If the signal goes back to 0, we know we're below the threshold for permanent damage. If this goes up, we know we're above the threshold for permanent damage. So we can very accurately determine that threshold, and then look how that threshold varies as we change the focusing, the numerical aperture basically, which is how tightly we focus the laser beam. So here are very tightly focusing microscope objectives, here are lenses, and notice that the more tightly we focus, the less the energy that we need in order to cause permanent damage. Ideally, we'd like to know not energy, but intensity. Well, intensity requires us to know what the aperture is, what the beam waste is at the focus. But remember, the beam focused inside glass, so we cannot easily measure inside there. However, the energy threshold and the intensity threshold are related by a very simple expression which only involves numerical aperture, the pulse duration, and the wavelength of the light, all of which we know. So we can just fit this, and out of it rolls the intensity threshold, which is a whopping, I emphasize, whopping 2 to the 7 times 10 to the 17 watt per square meter. What's the illumination of sunlight? 1,000 watt per square meter, so 14 orders of magnitude less. So the sun would have to be 14 orders of magnitude higher for those same type of effects to happen here. Luckily, we're very far from that, otherwise we'd have a lot of trouble here on Earth. We'd have a lot of trouble way before we get to this high. So this is for fused silica. We can now try different materials and repeat all of those series of measurements to determine what the threshold intensity is for other materials. And very quickly, we learned something really interesting, namely that that threshold intensity is not very material dependent. Here are some vastly different materials. Lithium fluoride, which has a band gap of 12 electron volts. We come in with a photon that has about 1.5 eV, so that means you need 8 photons to get across that band gap. Here, SF11, which is some form of fused silica of glass, only 3, so we only need 2 photons. Right? For 2 photons, the nonlinear term would be I squared. For 8 photons, it would be I to the 8th if it's purely nonlinear optics. So clearly, it's not just, it's an interplay between nonlinear optics to give the onset and then some linear absorption of the free electrons that causes this. Forget it if you don't understand what I just said. It's not that important. The key point is we can do this in just about any material, whether it's crystalline or non-crystalline, whether it's soft like a polymer or hard like diamond or fused silica or quartz. Everything is going to have a threshold that is roughly 2 to 4 times 10 to the 17 watt per square meter. It's a pretty universal phenomenon that we can use across a very wide range of transparent materials. In fact, there are no real outliers. Okay, so the thing to keep in mind from where we were when I first learned about biowork was we were good at studying the nonlinear interaction of light with transparent materials. We could exploit that interaction to disrupt matter inside the bulk, right? We wrote these little cavities deep inside the bulk of a piece of glass. And we can get this ablation, if you remember that curve, the tighter we focus, the lower the energy is needed at really very, very low energy. Well, about 10 years ago, Harvard was planning to build a new building, the Center for Nanoscale Studies, which was meant to be a building that was not owned by any specific department, but that was completely devoted to studying nanoscale phenomena, and that would foster the interaction between biologists, chemists, engineers, physicists, applied physicists, anybody who was doing work on the nanoscale. And in planning for that building, we invited people to talk. We had a series of talks where they would explain some problem in their field that dealt with nanoscale phenomena. And one of the speakers at that series was Don Ingbert, who I mentioned before, and he was basically talking about the cytoskeleton of cells. Let me explain to you a little bit about the cytoskeletons, and I will show some pictures later on. A cell looks like a pretty shapeless organism, but it has actually something that determines its structure, that is made up of two different types of materials. One is the actin fibers, and the other is a framework of microtubules, which are made from another polymeric substance. And Don Ingbert had developed a model for thinking about how an interplay between tensions and stresses, called tensegrity, determined the structure of cells. But one of the things he wanted to do was actually measure the viscoelastic properties of these actin filaments in cells. You can kill a cell, take the actin filament out, and then pull on it like a rubber band and determine what the elasticity is. But then you take the actin out of its ordinary biochemical environment, and it turns out that cells regulate the elasticity of these actin fibers depending on what they want to do. So ideally, you'd like to study it inside the cell, but how do you get inside the cell and actually measure the elasticity? I listened to it. I didn't understand half of what he said, but this whole idea about looking inside a cell appealed to me. So I went to him after my talk and I said, I think, we might be able to help you because we have a tool that would allow us to actually cut one of these fibers inside a living cell, and we could look at the elastic retraction. And a few weeks later was the beginning of a very long time collaboration. So the goal really was, how can we ablate material, remove material on the subcellular scale? So what do you need? You need sub-micrometer precision, right? You need, because the cell is not a big thing, maybe 20 to at most 50 microns. You don't want to do any damage to neighboring structures. Right? You don't want to accidentally puncture the nucleus or blow up some other part or damage the cell membrane. So you need to have very big spatial precision and very little collateral damage. Sounds like what I just told you about the glasses. And also it should be independent of the structure. If you want to do mitochondria or an actin fiber or microtubule, you should be able to do all of those without having to change your laser. So let's first talk briefly about the cytoskeleton. It's what determines the cell shape, and it also provides sort of a scaffold for the different organelles, and it's responsible for the motion of cells. I'll show you the motion of cells in just a moment. It's also responsible for cell division, and in fact, some of the work that was publicized last year in Cell magazine was work done in my group on looking at the role of the microtubules during cell division. I won't be talking about that today. So here are the actin fibers expressing green fluorescent protein on the left, and on the right are the microtubules, and you can see that they're slightly different morphology. We're going to focus initially on the actin fibers, which are the stiffer of the two fibers that determine a cell shape. And you can see that there are all these protrusions here, and these protrusions are ways for the cell to attach itself to the surrounding matrix. In this case, it's just a flat surface, and then pull on it, and that's what cells use to move around. Here's a little video of endothelial cells on a surface. This is under our microscopes, and you can see in this time-lapse movie that the cells basically move around. They don't stand still. They just move around, and they do that by having these protrusions and basically pulling on it and changing the biochemistry. Here you see a cell that is dividing. Did you see that? It was 1, now it's 2. Here's a cell that just had apoptosis, where it basically died. The 2 cells here have grown. I'm going to play this again so you can see it. So keep in mind, here there's a cell division, and here there's a dying cell. Whoops, there's another one that dies. So where's the one that is dividing? Let's see. Usually they let go of the substrate. Here. Is it this one? Yep. Both divided. Actually, the 2 turned into 4. Oh, there was another division. And here's the apoptosis, the cell death. So, I mean, if you just look through a microscope at a cell, you don't see this because you're unaware on an ordinary time scale. But your actual 150 trillion cells in your body are just as active as that on a slightly slower time scale than we usually deal with. So how do we do subcellular surgery? We use an epifluorescent microscope. So we take a sample that is fluorescently labeled, and we want, of course, to label the actin filaments. We come in with a UV lamp so that we can image the cells. There you see 2 endothelial cells. You can see there are 2 nuclei, and you can see these protrusions that are attached to the substrate. Then we bring in a femtosecond laser, and we basically cut the actin fibers that are under the crosshairs of our microscope. So here's an image, and you can see that we have translated these cells cutting through the actin fibers, and here we've made dots rather than lines. And if we look at a composite image of many planes, this is a confocal microscope, you can see that the cut is completely confined to the bulk of the cell. We've not damaged the front membrane or the back membrane that sits on the glass. That's very important because if you puncture a hole in the membrane of the cell, it dies. So how do we know we actually remove actin? Maybe the only thing we do is change the chemical properties of the green fluorescent protein, and it stops fluorescing. But the actin fiber is actually still there. So the way we did that was by looking at the nucleus of endothelial cells. This is an ordinary white light microscopy, but if we fluorescently label the cell, you can see the rest dark and the nucleus fluorescing. The reason we looked at the nucleus is because it happens to be the densest part of the microscope. Then we wrote 3 lines with 3 different powers of laser pulses. Pulses of 1.5 nanojoule, 1.8 nanojoule, and 2.3 nanojoule. By the way, how much is a nanojoule? Very, very, very little, right? I already told you, you drop something, it's fractions of a joule. I had to think really hard to come up with a daily life analog of nanojoule. If you take the kinetic energy of a flying fruit fly, it's about 10 nanojoule. I'm not talking about a fruit fly that hits the windshield of your car when you go at 50 miles an hour. No. If there was a fruit fly flying here and it bumped into your cheek, it would transfer on the order of 10 nanojoule. It's amazing that with just 1 nanojoule you can actually remove material, including glass or diamond. But you do that because you put all these photons together in so little space. Okay, so now we see darkened areas, but is it dark because we have basically bleached the fluorescence, or is it dark because we've removed material? So we went to a facility at Worcester Medical School where they can do electron microscopy, and we looked at the electron microscope image. If you remove material, it's more transparent to electrons, so it should be lighter. And notice that you can see the lines at 2.3 and 1.8, but not 1.5. So we know that at 1.5 we're bleaching, but at 1.8 we're actually removing material. So now we can actually put the ablation widths as determined by transmission electron microscopy and determined by fluorescence as a function of the pulse energy. We see that at the lowest energy, 1.5, we see nothing in TEM, but we see a cut in fluorescence. At higher energy, we see the same diameter in both approaches. So we really have 3 regimes. At very low regime, we see nothing. At very high energy, we see ablation, removal of material. And in between, we have this area where we actually bleach. And it turns out that this threshold, the threshold for ablation, is about 25% higher than the threshold for bleaching. And discovering that was really helpful because you can't afford to each time go to Worcester Medical School and do TEM, which is not an easy matter on living tissue. Now we know that if we slowly turn our laser on, at some points we're going to see bleaching. Then if we're about 30% higher, we can be guaranteed that we have ablation. So we use the bleaching as a, establishing that first threshold, and then from our measurements, many like this, we're able to determine that the next threshold is just 30% higher. So we have definitive proof of ablation. The ablation is as small as 100 nanometers, which is way beyond the resolution limit, but that is because it's nonlinear, so you actually end up narrowing that profile. The ablation threshold varies somewhat. It's slightly different from the nucleus than it is for an actin fiber, but in all of those cases, the ablation threshold is about 20–25% above the bleaching threshold. Okay, so can we do this now on live cells? Everything I showed you so far was done on fixed cells. Those are actually dead cells. So what about live cells? So we ended up taking cells where the mitochondria are fluorescent. The little spaghetti you see here are the mitochondria, very different from the illustration in the textbook that I showed you before. I was very surprised when I saw this. I had no idea what they actually looked like. And if we look at this area here, blown up in the top left, we see that there are 3 mitochondria that are very close to each other. These two are only about 500 nanometers away from each other. We're going to focus our laser beam right here at the tip of this arrow. And after nothing left, that one mitochondrion is completely gone, blown up, poof. But the other mitochondrion right next to it, just 500 nanometers away, has survived. This was already in itself a really interesting publication, even though it didn't get to the actin fibers, because in the biology community there was a lot of disagreement on whether mitochondria form a network or not. By doing this type of experiment, we were able to determine that the mitochondria are independent organelles. You can blow one away, it's not affecting the surrounding mitochondria. Let me show you a little movie that shows this happening in real time. So you're looking at the actual surgery as it happens. The movie is going to show the mitochondria. It's not going to show the laser pulse because the laser pulse isn't 800 nanometers, which is outside the range of the detector that we used. So here is here's the cell. This is the mitochondria that we're going to irradiate right here. Now it's happened. And notice that it pearls up and falls apart. There, boom, this has been hit. It falls apart into these little regions and then fade away. This is actual real-time nanosurgery in the, or subcellular surgery, I should say, in a living cell. How can we be sure that the cell survives? There are 2 ways of doing that. One is to look if the cell continues to divide. So we operate on these cells and then we put them in a medium where we can culture them and we wait and see if they divide. If they don't divide, then they're probably dead. If they divide, they are surviving. Another way is to look whether or not we puncture a hole in the membrane. And one way of testing that is by putting the cells in an ethidium bromide solution. Ethidium bromide is a dye that starts to fluoresce once it reacts with nuclear material. So the cell membrane is impermeable to ethidium bromide. So normally when you put a cell in ethidium bromide, the nucleus looks dark. If you were to puncture a hole in the membrane, the dye would go in and the nucleus would light up like a Christmas tree. So the first thing we did was focus on these mitochondria right here, this one, the one that's sort of curved, which is in the bulk of the laser, of the cell, pardon me. We fire our laser and we remove that one mitochondrion. The curved one is gone now, but notice that the nucleus is not lighting up. Notice, by the way, that the overall fluorescence went down a little bit, right? This was the previous image, this is this. That's because each fluorescent protein can only emit about 1,000 photons. So if you illuminate it with UV light, it will absorb, emit, absorb, emit, absorb, emit, but after about 1,000 emissions, it stops working. So it bleaches because of the exposure to UV light. So each time that we take a picture, we have to illuminate the cell for half a second or so, and you get some bleaching simply because of the act of making it fluoresce. But it has nothing to do with the femtosecond laser radiation. So next what we can do is we can move this target up to deliberately puncture a hole in the membrane and see what happens. Boom, the whole thing starts to fluoresce. So we know that we're at least, we can operate inside a cell without damaging the membrane. So can we now use this to probe the dynamics of the cytoskeleton? That's when we finally got to this question that Don Ingber had raised in his talk that I attended. So here is the actin fiber network. Here are the protrusions on which the cell pulls to move. This is the nucleus. And what we're going to do is we're going to focus our laser beam on this bundle of actin fibers. And after irradiating it, it pops like a rubber band and then retracts over time. This is over 10 seconds. Retracts like a rubber band. But is it actually retracting like a rubber band? Or is it just depolymerizing at the end, right? You could imagine cutting it and then the molecules just, go away. And it's not actually an elastic retraction. No, it's just a disintegration at the exposed ends because it's a polymer, so, it just falls apart there at the end. That's where this knowledge about bleaching and ablating came in really handy, because this is what we did. We first wrote a bleached marker. So we go below the ablation threshold, but above the bleaching threshold. So here, we've basically erased the fluorescence from this fiber. Now we translate our laser, and we're going to focus it here, but at a much higher intensity, and that's enough to ablate the material. So now it's going to retract. If it retracts, then this marker should move along with the motion of the ends. If it's depolymerization, then the marker should stay here, and this end should retract. And you know what? Look at that. The ends retract, and notice that the position of the marker moved, and you can even work out the ratios, and it works out beautifully. So now what we can do is we can take a little movie, and we can basically measure up all these frames and measure up the retraction, I'm getting tired here, the retraction as a function of time. And we can make a plot of retraction, the retraction distance as a function of time, and model this with a damped elastic retraction and determine this retraction time for it. So we can basically get the width of the original cut as well as the retraction time. And this is when it started to get really interesting, because now we're doing this in a living cell. As I said, cells can regulate the retraction. That's how they move around. And there are several inhibitors that can be injected in cells that change the elastic properties. So now we can actually see what the effect is of different biochemical molecules on the tension in these actin filaments. Here's, for example, a retraction that was measured after administering of this component called Y27. Don't ask me what it is, because I'm not a biologist, but it inhibits some of the myosin activity, and the myosin is what cells use to regulate the tension in the cell. This is an indirect blocker of the myosin activity. And here's a direct inhibitor of the myosin activity, which completely removes all of the tension. At that point, Don Ingber got so enthusiastic that he decided to buy his own femtosecond laser. Of course, that's very flattering, because that's when you realize that as a physicist you have an impact on the biological field, but at the same time it's the Achilles heel, because what was I going to continue to do? Luckily, that very same week, we had an applicant in the physics department who was interested in studying the origin of behavior as determined by neurons in a very simple organism. And I was listening to that talk, and I thought, I have the perfect tool. Here's what he was interested in. I have to tell you a little bit about neurons. Neurons are just like any other cells, except that they have dendrites and axons, and the signals come in through the dendrites, and that excites the cell body, which then sends a signal through the axon to the end of the axons, which then communicate with other neuron cells. But in and of themselves, apart from these axons and dendrites, they're cells like any other cell. In this little organism called C. elegans, which occurs everywhere in the soil all around this planet, it's a very simple organism, actually, and it's one of the biological model organisms. You know, the Drosophila, the fruit fly, yeast cells. I mean, going from small to large, yeast cells, C. elegans, Drosophila, you know, and eventually you get to mice and rats. I mean, they're called the model organisms. The genome was fully sequenced over 10 years ago. It's very easy to handle. Even a physicist can handle them. If you're interested in a specific C. elegans, you look up in the catalog, and then you call the group in Australia or New Zealand who happens to have this mutant, and you say, can you please send me some mutants? They put them in a FedEx envelope and ship them to you, and they're enough that survive the transit to make it, and the only thing that they eat is E. coli bacteria, so you just give them some E. coli. If you want to go on vacation, you put them in the refrigerator. They go into a state where hibernation, so you can go away and then take them out and they come alive again. It's absolutely miraculous. You don't need to have a background in biology. They're very small, 80 microns by 1 millimeter. They have about 1,000 cells, 302 neurons of those 1,000 cells. Not 301. Not 303, no, 302, all of them. And it's an invariant wiring diagram. It's just like, you know, a Toyota Corolla coming off the assembly line. They all have exactly the same wiring. You and I don't have the same wiring. In fact, any organism higher than C. elegans, even a Drosophila, does not have an invariant diagram, wiring diagram. But these are invariant, okay? You can. This neuron is connected to that neuron is connected to this neuron. You go to a website called Worm Atlas, and you can actually get a parts map. All cells are identified with this 3-letter code, and exactly which does what, which is connected to what. It's amazing. It's absolutely amazing. But the interesting thing is, just 302 neurons, which makes it a very intelligent little organism for its size, right? 1/3 of the cells are neurons. We can't claim that. We have very far fewer of our cells that are neurons. But that little network completely encodes the behavior of this little animal. It knows how to sense temperature. It knows how to sense chemistry because it needs to feed itself. It knows how to remember where food is, just how to walk. No, they don't walk with legs. They move around. Here they are on a Petri dish. And this Petri dish has a ring of glycerol, which is very bad for them because it reverses osmotic pressure. So they go, you'll see them crawl, and then they detect this glycerol, which is bad for them, and they turn around. Here we go. This is sped up by about a factor of 15. And each is about 1 millimeter long. So, you know, much smaller than on the screen projected there. Let's look real time close up to their locomotion, a little bit like a snake. Okay? There they go. And you know, you touch them with something hard and they go back because they feel the pressure. You put them on a plate that has a temperature gradient, 20 degree to 24 degree, and you put the food, the E. coli, at 23.1 degree. They learn very quickly that food is at 23.1 degree, and they memorize that temperature. You put the food at another temperature, they go to 23.1 degree, and they don't find the food. Eventually, they reprogram themselves to find the food at this new temperature. 0.1 degree memory of the temperature they have. It's pretty amazing. Okay, very quickly about the life cycle. They start as eggs in another. They're hermaphrodites. There are no female and males. Then the eggs, after 2.5 hours that they're formed inside the C. elegans, are exuderode. 9 hours later, the first larval stage. 12 hours after that, the second larval stage. 8 hours that, the third one. 8 hours after that, the fourth one. And then we have an adult. and just a little bit later, 8 hours later, they start to form eggs. So that cycle is fantastic, right? Because you can go through many generations very quickly, which means if you need to develop a mutant species, you can do many cycles very quickly. There's this interesting thing. If you deprive them of food or make it too cold for them to survive, they go into this dour state where they still crawl around, but their mouth is shut. And they basically work on internal energy reserves for 4 months in this sort of hibernation stage. They still move around, it's amazing, without any food. And then when they detect that there is food again, or that it's not too crowded, or that the temperature is better, they jump all these other larval stages, 2 and 3, straight to 4, and then go into an adult life. So when I heard this talk, this all sounded very different. I'd never heard something like this. And he told me how he was doing his experiment. His experiment was basically to look at the catalog of C. elegans and find a mutant that was missing specific neurons. And then looking at how the absence of that neuron affected the behavior in order to infer what the role of the neuron is in the behavior. So I thought, wow, I mean, what if you can't find the right mutant? Or what if that mutant has other changes that have escaped your, it's not just that one neuron that is missing, but there are other things that are different about it. Wouldn't it be much cleaner if you could go in with our laser and just cut a connection between 2 neurons? It's like snipping a wire in an electric circuit diagram. So I went up to him after my talk. He was an applicant for a junior faculty candidate. I don't know if I told you that. And we hired him. And 2 weeks later, we hadn't even made up our mind yet, he and I were collaborating and doing the first experiment. So the idea is this, you know, don't go for mutants, just dissect the neuron, or even better, the dendrite or the axon between the neurons, and then look at the behavior and infer the role of the neuron. So the first neuron we looked at was the ASH neuron. As I said, each cell is identified by 3-letter code, so I don't think there's any meaning to it. Those are responsible for chemical sensing. There are 2 of them, one on the left, one on the right. And they basically go all the way to the mouth of that little animal. It really has a mouth, which is it's a tubular organism, with a mouth in the front and then a gut that extends all the way to the back. Now, it makes sense that they are at the front. It makes sense that you have 2, right? You want redundancy, so that if you lose one, you can still use the other. And also, you want it all the way in the front, because you don't want the animal to walk into glycerol, let's say, and then only realize that it's walked into glycerol after ingesting the glycerol. So that makes a lot of sense to put that on the outside. So this is a microscopy image of, you can sort of see the mouth here and the gut that goes all the way to the back. We're going to look at the front. This is a diagram of the front. Here are these 2 ASH neurons, and this is an axon ring. The 2 are connected. Their 2 axons are connected, and that connects to other neurons that are not shown here. And then the dendrite go all the way to the front, one on each side of the animal. So there's a left cell body, right cell body. Here's the axon ring, and there are the 2 dendrites, one on each side. Now, that would be easy if there was just one dendrite, but it also needs to feel pressure, right? It needs to know whether or not it walks into a wall or something else, or it needs to know whether it's hot or cold. So there's not just one dendrite, there are many dendrites, the whole cable bundle that goes towards the front. So the first thing you need to do is know which of those dendrites you want to have. So you basically insert the gene for green fluorescent protein in the gene sequence for the ASH neuron, so that all ASH neurons will start to fluoresce. Here's, for example, here they are made visible. You can see the axon ring here and the 2 dendrites. Here's a little movie that shows the fluorescently labeled, so this is an ordinary light. When you turn on the UV light, you can see the neurons light up. And you can see it in an egg that is inside the body. You can see some also light up. There they are. They light up, right? So we can actually make them visible. Of course, the darn animal is moving around. So you have to target it too. I have good students, but not that good that I can hit with a femtosecond pulse. So we anesthetize the the worms so that they basically are unconscious and not moving, and then we do the surgery. Okay, so here's the cut of a dendrite. We basically put it under the crosshairs, fire our laser, and you see a similar type of retraction. So no more electrical or chemical signal can travel down that dendrite. But the problem is it's not just the ASH neuron. As I said before, there's the ASK neuron, the AUA neuron, all responsible for different things, the ASI neuron. So you need exquisite precision because they're all traveling towards the front, right? If you cut all of the dendrites and then you look at the behavior, you don't know which one has caused the change in that behavior. So what you can do is you can label them all fluorescently. Here they are. There are actually 5. And then identify the one you want to cut, and then see if you can cut just this one but not the other ones. So let's cut the temperature sensor and see if it can still detect chemistry. Or let's detect the chemical sensor and see if it can still detect temperature. Once we've done all of that, we were ready to do our experiments. Also, we want to see how the cut evolves. Does it regenerate? Right? I mean, some neurons regenerate, others do not. So we revive the worm, which is very easy. We just rinse it underwater, and then the anesthetic wears off. And then we re-image it, and we can go to much better microscope. So here you can see the axon ring, the 2 cell bodies, and you can see the dendrites, and you can see how the cut survives one day later to make sure that there are still no electrical signal can travel. So the first thing we did was look at this chemical sensing. So we put these worms in this glycerol ring, and we look how many worms escape, right? So over time. So one is, you see, when we prepare these worms for surgery, there are always a few that are not lying correctly on the plate, but we still anesthetize them, do everything else. So those are our control. We take the worms that we've prepared for surgery, they've been anesthetized, but we've not actually done the surgery. Then we revive them, and we look at how many escape as a function of time, and because they still have their sensors, they don't escape because they notice the glycerol. You can look for a mutant that is missing that ASH neuron. So you just have a few of those shipped to you, and you see how many of those escape in 5 minutes from that glycerol ring. And you find that about 85% or so escape. Right? So if they don't have ASH neurons, 85% of them escape in 5 minutes from that ring. So now let's look what happens when we do the surgery. If we cut just one dendrite, no difference. They're still all at 0 because they still have the other dendrite to detect. If we cut 2, we find the same 85%, but now we can also look if there's regeneration because we're going to look at it over time. So right after surgery, it's right on here, but what is going to happen for longer times, as you can see here, it doesn't vary, which means there is no regeneration. We've shut off that sensor for good. After doing that, we turn to the AFD neurons because there's some interesting physics there. The AFD neurons are the temperature sensors. They also have these 2 dendrites. Why does it have those 2 dendrites? Right, I mean, if we want to know the temperature outside here, we need to put the temperature probe outside because we can't measure the temperature here outside. But when you are the size of C. elegans, it doesn't matter where you measure it. The cell body is always in equilibrium with the tip of the animal, right? It's so small, it's all in thermal equilibrium. So you don't need to measure the temperature outside. You can measure the temperature inside. So what's the role of those dendrites? Where does the temperature sensation happen? Well, that's something we can do not by looking at mutants, but only by looking at the surgery, because we can cut the dendrite at different spots, close to the tip, far away from the tip, and see where the temperature sensation ends. So here's another picture of this AFD neuron expressing green fluorescent protein, the 2 cell bodies, the axon ring, the 2 dendrites. Here's actually an egg. Here are 2 other eggs. I don't know if you can see them on the screen. No, you can't. But you can't see this egg that has started to, fabricate neurons that already are expressing this green fluorescent protein. And now we can cut these AFD, and we can look how it affects the temperature sensation. And to do that, we basically put the C. elegans in a little droplet that is hanging from a glass slide. So the C. elegans can swim, but it swims against gravity. It doesn't know it's a small droplet. It can swim forever, right? Swim, 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 because it basically drops back down under gravity. So for the C. elegans, this is a C rather than a droplet. We can now change the temperature. We can oscillate the temperature. If C. elegans detects that it's getting too hot, what does it do? It turns around. And when it turns, it basically puts the nose close to the, you can see it go like this, and then it curls, and then crawls again. So you can measure the number of turns it takes as you change the temperature and find out whether it still can detect temperature or not. We have a video camera that looks at the C. elegans from the bottom. So this is a C. elegans swimming, not turning. It basically goes straight because it's swimming against gravity. We're looking up. Here on the right is a worm that we have operated, and unfortunately the operation went not quite right. We hit it a little bit too hard, but I'm using this video because it's clear that the behavior is so different. It's not very happy, as you can see. It actually turns continuously. But we can basically, and I'm leaving out many quantitative detail here, basically correlate the surgery to the behavior. And what we determined was that the temperature occurs at the very tip. There's no physical reason for that. There is no physical reason for that. But somehow, probably because temperature and chemical sensation are related. As you know, mint is something we somehow associate with cold, and peppers with hot. So even in our minds, taste and temperature are related. And the sensor and the tip of both the chemical and temperature sensor are very closely related. So it must have something to do with the evolution of those two sensations. But this was the first conclusive evidence that the sensation actually happened at the tip. Well, by this time, R.V. Samuel was a junior faculty at Harvard, and he got really, really excited, and he spent his startup fund on buying his own femtosecond laser. And I thought, he knows everything about C. elegans, and now he knows everything about femtosecond lasers too. What am I going to do? The interesting thing is that the story repeated itself 2 more times, first with cell division and then with self-transfection. So I'm still in the area, and each time I think, there goes my experiment, and that somebody else is adopting it. There's another opportunity. But I hope I've shown you that this is a great tool to manipulate the machinery of life and unlock some of those amazing mysteries of how it works. If you want a copy of my slides, they're on my website, ready for downloading. You don't need to write this down. Just remember my last name, M-A-Z-U-R, enter it in Google, and then hit this I'm feeling lucky button, and then in one more click, you'll have a copy of the slides. Thank you very much.
I can take a couple questions, but before we do, I'd like to point out some irony here is that my first